So I'm going to move to the opposite end of the spectrum and talk about receptor pharmacology, specifically a concept known as G protein biased agonism, and how this might be applied at the GLP-1 receptor, which, as I'm sure you all know, is an important target in type 2 diabetes and obesity. So here's my disclosure slide. So my lab is interested in understanding the factors which can influence G protein coupled receptor or GPCR signaling, and the consequences that this can have in type 2 diabetes and obesity. And I'm going to focus particularly on this topic here, biased agonism. But I'm also interested in the impact of missense variants in the target receptors that we study, and other things like the local membrane environment in which these receptors are sitting. And we use a variety of different in vitro and in vivo approaches to measure these differences, with the aspiration to try and translate these findings into humans in the future. So just a little bit of undergraduate pharmacology. Many of you will recall that agonists can be divided into either full agonists or partial agonists. And a partial agonist, even if you give a high dose, will only reach a sort of submaximal response, as shown here. But actually, of course, these receptors are coupled to a variety of different intracellular proteins. So we have, for example, the G proteins, as shown here. They are primarily responsible for intracellular signaling. And then we also have the beta-arrestins, which have a number of roles, but one of those is to switch the receptor off, desensitize it, maybe produce tachyphylaxis. And that's really important physiologically. We need to have ways of switching off hormone and neurotransmitter signaling for physiology. But for a drug, that might not be ideal because you want your drug to have a long-lasting effect. So we've been quite interested in the idea of G protein biased agonism. So in this situation, we have specifically designed ligands which activate the same receptor. They still produce G protein dependent signaling, but they can avoid beta-arrestin mediated desensitization. And so the idea here would be that we can promote longer term signaling by avoiding these switch off mechanisms. I'm just going to move. I'm not sure if this thing on the side is in front of the screen, but hopefully you can see my entire slides. Anyway, I don't seem to be able to move it. Okay, so that's an interesting pharmacological concept. Why might it be relevant therapeutically in the context of diabetes and obesity? Well, you'll be aware that there's a number of GLP-1 receptor agonists available on the market. Liraglutide, dulaglutide, semaglutide, and a new one, danuglipron, which is currently in phase 3. It's an oral GLP-1. It's quite an interesting one. These are all considered as balanced GLP-1 receptor agonists, so they're not biased. But what we also have are some new molecules, for example, tirzepatide. This is actually a GLP-1 GIP receptor co-agonist. So it hits two receptors, but at the GLP-1 receptor, it has quite pronounced bias in favor of the G protein pathway, and now approved for type 2 diabetes, and under consideration for obesity at present. Another example currently in phase 3, orforglipron. Again, this is an oral GLP-1. It's not a peptide, so sort of like a small molecule, although not quite. And again, this has got really quite strong bias in favor of G protein signaling. I think it's very difficult to directly compare these kind of ligands, but I think overall the data suggests that these ones might be a bit better than these ones. Now, the problem is we don't know if that's due to the biased signaling, which they clearly do show, or whether it's something else. Obviously, tirzepatide as a co-agonist acting at a second receptor, that's clearly a potential reason why it may be better than a monoagonist such as semaglutide. Or it could be the bias, which would be sort of interesting for me. And the problem is, because these molecules have all got different stabilities, different pharmacokinetic profiles, different dosing regimens. You can't directly compare them on the basis of the available clinical data, at least if you want to infer mechanisms. So that's something that we've been working on for a little while now. So one of our aims has been to develop oppositely biased GLP-1 receptor agonist molecules, which have got equivalent stability and equivalent pharmacokinetics as well. So we take those differences out of the equation, and so we can just look at any sort of functional consequences which could be attributed to this biased signaling. And then we sort of test these in cell models, in preclinical in vivo models, disease models, and then eventually we wish to do studies in humans as well. So to give you an example of a pair of good examples here. These are a couple of tool compounds that we've been working with for a while now. They are peptides. They're heavily based on exendin 4 or exenatide by-ETA. So you may know it, existing type 2 diabetes drug. And the only difference is that they have amino acid substitutions at or close to the N-terminus, as highlighted with the sort of colored circle there. And yeah, because they're so similar in all other ways, they have the same stability, the same pharmacokinetic profiles. And so we think that we can use these to tease out any effects, downstream effects that they have, which it might be attributable to bias. And as an example, so this is a sort of an in vitro signaling assay. On the left, we have a dose response looking at G protein signaling, cyclic AMP. And on the right, we have beta-arrestin recruitment. And you can see that both ligands have the same maximum response for the G protein pathway, but for the beta-arrestin response, then the F1 ligand basically just does nothing. So some people might refer to this as a perfect degree of bias between G protein and beta-arrestin signaling. I think it's worth just flagging that there is also a difference in the G protein pathway, which is that the F1 drug has reduced signaling potency. So in vitro, you need to give higher doses to get the same effect from this sort of left-right shift here. But anyway, so what might be the impact of this different pharmacological profile? So showing this image again, then because the arrestin, one of their primary roles is to switch the receptor off, by avoiding that response, then we might anticipate that the F1 drug would produce longer lasting signaling effects by avoiding desensitization, tachyphylaxis. And indeed, this is what we see in vivo experiments in mice. So here, what we have is mice which have been treated with these biased agonists or derivatives thereof, or a control. And what we can see is that over time, after a single injection, the mice which have received the F1 drug can eat less. And that tends to diverge after about sort of 12 hours plus. And then when we extend this over a longer treatment period, 2 weeks, then we see that line continues to diverge, and this leads to greater weight loss. And this, I think, is quite interesting because, as I showed on the previous slide, this biased agonist drug has actually got quite low signaling potency. So, it's something that probably would have been discarded in a traditional drug discovery program because it has low signaling potency, but actually it works very well in a physiological system. So the question is, does this matter in humans? Well, the answer is we don't know yet. There is quite an interesting paper from Ewan Pearson's group, which analyzed the impact of mutations, loss of function mutations in the beta-arrestin 1 gene. And in the context of GLP-1 receptor agonist trials, these mutations, shown in blue, led to a greater response to these drugs in terms of the ability to lower blood glucose. So I think there are some clues there. We do need larger scale pharmacogenomic data to clarify the role of beta arrestins, the role of bias signaling in the therapeutic response to GLP-1. And I think we also need to be doing a sort of direct physiological comparison of the kind of ligands that I showed on the previous slide, where we know that they have the same pharmacokinetics, and we can compare how they act physiologically in humans. And then the final point I wanted to make is that, well, you may well be aware that there's quite a lot of missense coding variation in all genes. And certainly for the GLP-1 receptor, around 2/3 of people have at least one missense coding variant. Probably most of those are fairly benign, but there are some which are potentially more impactful. And we were interested to know whether this might, the functional impact of these kind of variants might have ligand-specific effects. So will they differentially impact the ability of the receptor to respond to different drugs? And so, without going into the detail, the bottom line was that when we compared 3 sort of clinically relevant GLP-1 receptor agonists, exenatide, semaglutide, and tirzepatide, then, for example, this variant here, which is an important one physiologically, showed quite a marked discrepancy in terms of its ability to respond to tirzepatide versus exenatide. And of interest to me, tirzepatide is a biased agonist. So yeah, it may be that this applies beyond tirzepatide and could apply to other biased agonist ligands as well. So just to finish up, in my opinion, biased agonism is a potential route to improving the therapeutic targeting of G protein coupled receptors, including potentially the GLP-1 receptor. Tirzepatide is a good example of a G protein biased GLP-1. We don't know if its beneficial effects are due to the biased signaling or whether it's primarily due to the effects of its GIP receptor activating component. And these kind of questions still need to be addressed in a proper human study. And then finally, so there's a lot of variation in these kind of GPCRs. The pharmacogenomic impacts of that are not currently known. And I think that will hopefully come out at some point in the future through large-scale pharmacogenomic data from the trials which are ongoing. And here are the number of people who've contributed to this work. Thank you very much.